Hey everyone, and welcome. Today, we're exploring an explainer on something absolutely fundamental to life as we know it, protein structure prediction. Imagine you're trying to guess the exact shape of a highly complex 3D origami sculpture, but you're only allowed to look at an unfolded, flat piece of paper. Well, in the world of bioinformatics, scientists are basically doing exactly that. They're figuring out the precise three-dimensional structure of a protein from nothing but its raw amino acid sequence. It sounds a lot like science fiction, I know, but it's happening right now and it's driving some incredible breakthroughs. So let's just dive right into this and see how scientists are literally decoding the architecture of life.
Here's our roadmap for today's explainer. We'll start with why protein shape actually matters, look at the 4 levels of structure, break down how we predict those structures, talk about the massive AlphaFold AI revolution, explore real-world applications, and finally wrap up with the future of bioinformatics.
All right, part 1, why protein shape matters.
So why do proteins matter so much? You probably already know they're made up of basic units called amino acids. But here's the thing. A protein isn't just some floppy string randomly floating around inside your cells. No, for a protein to actually do its job, whether that's digesting your food, fighting off a nasty virus, or carrying oxygen through your blood, it absolutely has to fold into a highly specific, three-dimensional shape. Its entire biological function is totally dictated by this final physical form.
And that brings us to the golden rule of structural biology. Sequence dictates structure, and structure dictates function. It's that simple, but it's incredibly powerful. The specific sequence of amino acids naturally forces the protein to fold in a certain way, creating a very specific 3D structure. And that structure? Well, that determines what the protein can actually do. The stakes here are just massive, because even the tiniest change or mutation in that structure can drastically alter how a protein works in the human body, which is what sometimes leads to severe diseases.
Okay, let's move right into part 2, the 4 levels of structure.
It all kicks off with the primary structure. You can think of this as the foundational blueprint. It's basically just the linear sequence of amino acids strung together in a chain, kind of like beads on a necklace. And the exact order of those beads isn't random at all. It's directly determined by your underlying gene sequence. This simple string contains all the necessary instructions for the incredibly complex folding that happens next.
So from that linear chain, the protein starts to fold into its secondary structure. It forms this basic scaffolding, things like spiraling alpha helices or folded beta sheets, and those are held together by hydrogen bonds. But we don't stop there. It then folds even further into its tertiary structure, which is a really complex 3D shape stabilized by a whole mix of hydrophobic interactions, disulfide bonds, and ionic bonds. Finally, some of the more complex proteins take it one step further to the quaternary structure. This is essentially the arrangement of multiple protein subunits all working together as one giant machine. A perfect example of this is hemoglobin, which is the protein responsible for carrying oxygen in your blood.
Which leads us nicely into part 3, how we predict structures. Solving this giant 3D puzzle requires a highly systematic, basically a 6-step pipeline. First up, researchers have to obtain the raw amino acid sequence. Second, they run it through a search tool like BLAST to hunt for similar already known structures in existing databases. Third, based on whatever they find, they choose a specific prediction method. Fourth, the computer finally builds the 3D model. Fifth, they refine the structure to make sure all the underlying physics and chemistry actually make sense. And finally, step 6 is validating the overall quality of the model before anybody uses it for actual research. Now, when it comes to step 3, actually predicting the model, researchers basically have 3 main options, depending on what information they're starting with. The first is homology, or comparative modeling. The principle here is pretty straightforward. If 2 proteins have similar sequences, they're probably going to have similar structures. For this, researchers use really powerful tools like Swiss model or Modeller. But what if you don't have a similar known structure? Well, then you pivot to ab initio or de novo modeling. This predicts the structure entirely from scratch, using sheer physical and chemical principles. There's no template involved at all, which makes it incredibly compute-intensive. Lastly, we have threading, also known as fold recognition. This method tries to fit the new sequence onto known structural folds. It's super useful when no close homologue exists, but researchers suspect the overall shape might match something we've seen before.
All right, let's jump into part 4, the AlphaFold AI revolution. Modern bioinformatics has been totally transformed by AI systems, and the absolute prime example of this is AlphaFold. AlphaFold predicts protein structures with near experimental accuracy. It has, literally overnight, revolutionized structural biology.
By completely bypassing years and years of painstaking, slow lab work, this AI is helping scientists understand entirely unknown proteins at speeds that are just breathtaking.
But, an AI is really only as good as the data it learns from. It heavily relies on two foundational databases. First, there's the Protein Data Bank, or PDB, which stores actual, experimentally determined 3D structures. Then there's UniProt, which houses vast catalogs of protein sequences along with their functional information. Together, these two databases provide the essential raw material that makes all this computational prediction possible in the first place.
So what does this mean outside the lab? Part 5. Real-world applications.
The real-world applications here are honestly staggering. In drug discovery, knowing the 3D shape of a protein allows scientists to find the precise physical pockets where a new medicine can snap in, almost like a key fitting into a lock. In disease research, we can visually spot exactly how a cancer-related protein goes rogue. It allows enzyme engineers to tweak proteins to make them more stable for industrial use, and it helps immunologists design exact targets for new vaccines. Broadly speaking, it is supercharging all sorts of biotechnology, including entirely new synthetic biology applications.
Now, while this technology is incredibly powerful, we've got to look objectively at where the field stands today. The advantages are crystal clear. It radically speeds up drug development and massively reduces the need for really expensive slow lab experiments. It helps us study unknown proteins fast, but it's not flawless yet. It still struggles with accuracy for certain highly complex proteins. Membrane proteins are notoriously difficult, for example. And because real proteins naturally flex and move around, modeling that dynamic biological flexibility is still incredibly hard and requires a massive amount of computational power.
Which brings us to our final section, Part 6, the future of bioinformatics. This field is moving at lightning speed right now. Future AI models are actively aiming to overcome the hurdles we just talked about, pushing us closer and closer to real-time protein folding predictions. Imagine being able to watch molecular movements exactly as they happen. As this tech integrates directly with automated drug design systems, it's going to unlock true personalized medicine, meaning treatments tailored to the exact mutated structures inside an individual patient. Plus, we're going to see much better modeling of how multiple different proteins interact with each other in complex cellular environments.
We've seen how decoding these functional 3D structures from simple sequences of amino acids has opened the ultimate door for biology, medicine, and biotech. It truly is the architecture of life. So I want to leave you with this thought-provoking question as these computational models keep getting faster and more accurate. If AI can predict the architecture of life in real time, what will we cure next? Thank you so much for joining me on this explainer, and definitely keep questioning the science around you.